All right, welcome everybody to lecture number 4, which is protein structure part 1. So this will be a 2-part lecture series just on protein structure. So today we'll cover primary and secondary protein structure. And first we'll talk about primary protein structure, which is how amino acids are joined together using peptide bonds into a long polypeptide chain. And we'll talk a little bit about the limitations on the movement of that chain in three-dimensional space. We'll then move on to a discussion on secondary structure, which is how that polypeptide chain will fold into regular conformations in a local space. So we'll talk about alpha helices, beta sheets, as well as turns and loops. So I hope you guys all enjoy the lecture. So let's launch right in. Lecture number 4, protein structure, part 1. So we'll start our lecture today talking about the 4 levels of protein structure. So the 4 levels of protein structure are primary, secondary, tertiary, and quaternary. So today we'll be talking about primary and secondary structure, and then we'll talk about tertiary and quaternary structure in the next lecture. So primary structure is the sequence of amino acid residues in a protein. So amino acids are going to be linked by peptide bonds into a polypeptide chain, and the primary structure is the precise sequence of amino acids in that polypeptide chain. Now, the sequence of amino acids in that polypeptide chain will ultimately dictate the overall protein structure. Now, secondary structure is the local folding of that polypeptide chain into regular structures. So this is dictated by the backbone hydrogen bonds of those amino acids, so not the side chain. Secondary structure is dictated by the backbone hydrogen bonds. And this causes local folding into regular structures like alpha helices, beta sheets, and then turns and loops. There's also tertiary structure, where a globular protein will fold into a compact structure. So this is the folding of that secondary structure into a compact structure. So this is actually dictated by the side chains. So hydrophobic side chains will generally be found on the interior of a folded protein, and hydrophilic residues will be on the exterior. And then the interactions between those side chains and between the side chains and the backbone will also dictate that tertiary structure folding. And then quaternary structure is where multiple folded polypeptide chains will interact to form a complete protein. So this will be caused by protein-protein interactions between identical subunits or identical polypeptide chains or different polypeptide chains to assemble the full protein with quaternary structure. So not all proteins have quaternary structure, but many, many do. So those are the 4 levels of protein structure, and today we'll talk about primary structure and secondary structure. So as I said, the primary structure of a protein is going to be caused by linking amino acids into a specific sequence by peptide bonds. So these amino acid residues are linked by peptide bonds, and this linkage is an endergonic reaction called a condensation reaction that requires an input of energy. So here you see we have 2 amino acids, and then they're linked by this peptide bond between the carboxyl group here and the amino group here. And that releases a molecule of water, which is why it's a condensation reaction. And it requires the input of energy, like I said. So peptide bonds are very stable. So without a catalyst, they'll remain intact for 1,000 years. So you could see that these peptide linkages cause very stable assembly of amino acids into polypeptide chains that are linked by this peptide bond right here. So as I said, these peptide bonds between amino acid residues are going to be combined and lengthened to form a polypeptide chain. So polypeptide chains are both synthesized and written from the amino terminus to the carboxy terminus. So this is the way they're synthesized in the cell, and it's also the way you would write them when writing out a polypeptide chain. So always from amino terminal residue to a carboxy terminal residue. And so the polypeptide chain above here would be written as tyrosine, glycine, glycine, phenylalanine leucine, or YGGFL. So if you switch that, if it was leucine, phenylalanine, glycine, glycine, tyrosine, that would be a completely different polypeptide chain with different chemical properties. So peptide bonds are synthesized and written from amino to carboxy terminus. Now, there's two features of these polypeptide chains that I'd like to bring your attention to. One is that they consist of a constant backbone, which would be called the main chain, with variable R groups, which would be the side chains. So here you see we have the amino nitrogen, then the alpha carbon, and then the carboxy carbon, and then the next amino nitrogen, and so on and so forth. So this would make up the main chain that's linked by those peptide bonds. And then number two is that these potentially bulky side chains here tend to orient themselves away from adjacent side chains on that polypeptide chain. So you can see R1 here is oriented above and towards us. R2 tends to orient below and away from us in this figure. R3 above and towards us, and so on and so forth. So there's that link between the main chain here, and then the side chains stick out away from the main chain and tend to orient away from adjacent side chains because these are the bulkier groups on the polypeptide chain. Now, some other points. So most polypeptides consist of 50 to 2,000 amino acid residues. And the mean amino acid has a mass of 110 grams per mole. So that would make the average polypeptide about 5,500 to 22,000 grams per mole. And protein mass, we'll probably discuss that, or that'll probably come up every once in a while in this course. So protein mass is measured in daltons or kilodaltons. So most proteins are between 5.5 to 220 kilodaltons. So that's the average length of polypeptide bonds. So the largest protein known is a protein called titin that's involved in your muscles, and that's 27,000 amino acids long, but that's definitely an outlier. So most polypeptide chains will be in this 50 to 2,000 amino acid residue range. Now, another feature of peptide bonds are that they are basically planar in this polypeptide chain because they're a bit conformationally restricted. So the peptide bond, as you can see here between this carbon and this nitrogen, has resonance structure. So it's basically a partial double bond. So you can see these electrons here on this oxygen can come up here, and then the atoms on the nitrogen can come here to form a partial double bond. And this causes the peptide bond to be conformationally restricted, so it limits the movement about this particular bond. And you can sort of see that demonstrated down here. You can almost think of it as planar, that the double bond character causes it to be restricted on how those two atoms that form the peptide bond can actually move about that bond. And so the peptide bond is limited to a cis or a trans conformation. So here you can see a depiction of both the trans and the cis bond here. And the trans bond is much more favorable due to the steric hindrances from the R groups when they're in the cis bond. So basically, the peptide bond can only be in one or another conformation because it's got that planar sort of structure caused by that resonance structure. And the trans orientation is much more common and more favorable because you can see that these R groups, which are represented by the green spheres here, the green atoms, are oriented away from each other in the trans orientation, but oriented towards each other in the cis orientation. So just to review, so peptide bonds have planar characteristics based on the resonance structure, and this causes them to be conformationally restricted, and the trans conformation is much more favorable thermodynamically. So although polypeptide chains are conformationally restricted about their peptide bonds, which are the bonds located right here, The bonds connecting to the alpha carbon are able to freely rotate. So these are called phi and psi bonds. And this is what allows the protein to adopt many different conformations and actually fold into its three-dimensional structure. So although that peptide bond is conformationally restricted and planar, the phi and psi bonds that connect to the alpha carbon are freely able to rotate. So that's how you're actually able to get that overall folding that gives the protein its final structure. And so that rotation about these phi and psi bonds were actually investigated by this scientist named Ramachandran, and he developed what's called a Ramachandran plot. So he looked at actual protein structures and determined what angles about those phi and psi bonds were actually possible in proteins without steric hindrances. So without these bulky side chains bumping into each other or having sort of these steric hindrances that cause them to, interact or repel each other, or just not allowing different atoms to occupy the same space at the same time, because that's impossible. So he determined that there are very specific angles of these phi and psi bonds here that line up with this plot, where most phi and psi bonds are going to have conformations that correspond to specific regions on this Ramachandran plot, especially these two regions right here. So while polypeptide chains can rotate around their phi and psi bonds, there's definitely some conformations that are much more thermodynamically stable than others, and this Ramachandran plot is able to illustrate that. So if you're determining the structure, the three-dimensional structure of a new protein, you would always make sure that each of those individual amino residues on that polypeptide chain, their phi and psi bonds, conform to what's to be expected with this Ramachandran plot. And if there's any outliers, that probably has to do with some sort of local thermodynamics or physics or some local interactions that are causing it to be outside of these favorable parts of this Ramachandran plot. So now that we have a good understanding of protein primary structure, which is with these amino acids linked by these peptide bonds into a polypeptide chain that has specific ways in which it can rotate, now we can move on to protein secondary structure. So secondary structure describes the local folding of that polypeptide chain into these regular structures. So there are three types of secondary structure that you'll find in proteins. So number one is alpha helices, two, beta sheets, and then loops and turns that are going to sort of connect these alpha helices and beta sheets. So the first secondary structure that we're going to talk about are alpha helices. So alpha helices are very stable, and they form these rod or ribbon-like helical structures that are stabilized by hydrogen bonds in the polypeptide backbone. So this is very important to remember. So alpha helices are formed by interactions by hydrogen bonds in the polypeptide backbones. So the side chains are not what form alpha helices. So the side chains extend outward perpendicularly from these alpha helices. So there's some depictions here. You can see it's depicted as sort of this ribbon or rod-like structure, and it has helical characteristics to it. And then the hydrogen bonds between these amino groups and these carboxyl groups are going to stabilize that helix. So you can sort of see here. So this works on the principle that the oxygen of the carboxyl groups on the backbone and the hydrogen on the amino groups on the backbone are going to form hydrogen bonds. So that oxygen is going to be a hydrogen bond acceptor, and that hydrogen from the amino group is going to be the hydrogen bond donor. So there's a depiction right here. You can see in the helix itself, you can see these hydrogen bonds stabilizing the helix. You can also see it here on this depiction planar planarly. So this oxygen is going to hydrogen bond with this hydrogen from the amino group, which is 1, 2, 3, 4 amino acid residues further down the helix. And you'll see the same thing here with this oxygen from this carboxyl group onto this hydrogen in the amino group. So alpha helices are these rod-like, ribbon-like structures that are stabilized by hydrogen bonds in the polypeptide backbone. And then that allows the side chains to stick out perpendicularly from that alpha helix, which is also thermodynamically stable because these potentially bulky side chains are sort of able to stay away from each other on the polypeptide, on the alpha helix. And so there's a few points or a few features about alpha helices that I would like to draw your attention to. So number one is that there are about 3.6 residues per helical turn in these alpha helices. So you can see if this is a side chain from one residue right here, you have another residue right here, second residue right here, and then this third residue is not quite one full helical turn. And then the fourth residue here is a little bit more than one helical turn. So there's 3.6 residues per helical turn. Another point is that essentially all alpha helices in proteins are right-handed, so they turn in a clockwise direction. So you will find some left-handed helices, and they are actually accounted for on that Ramachandran plot, but right-handed helices are a bit more thermodynamically stable, and so they're much more common in proteins. Another point is that some amino acids are not accommodated well in alpha helices, so they'll actually cause the alpha helix to sort of break and turn into something else, like a loop or a turn or something else. So branched hydrophobic amino acids are not accommodated well in alpha helices, so that would be valine, isoleucine, and leucine. And this is due to steric clashes between those really bulky hydrophobic groups. Serine, asparagine, and aspartate, those are also not accommodated well in alpha helices, and that's because they have polar characteristics and they all have a short chain, side chain. So this actually causes them to compete with hydrogen bondings from the main chain. So those aren't accommodated very well either. And then also proline, which is due to its ring structure and its lack of an NH bond, hydrogen bond donor. Because if you remember, it has that ring structure where the side chain actually forms a covalent bond with the amino group. So now that amino group isn't able to be as good of a hydrogen bond donor. And so although all these amino acids, I'm sure if you look at all the protein structures, you will find each of them on alpha helices. They're just way less common because they're not accommodated well because of either steric clashes in the terms for valine, isoleucine, and leucine, hydrogen bond competition from serine, asparagine, and aspartate, and then proline due to its conformational restrictions based on its ring structure and its lack of a hydrogen bonding group. So a few more points about alpha helices because they're so important. Alpha helices are depicted graphically as either these ribbons, as you can see here. That's probably the more common depiction, or rods or cylinders, as you can see right here. So when you see a protein or an image, a representation of a protein structure, it's either going to have these rods or ribbons, which indicates that there's an alpha helix at that position. Proteins can be from 0 to 100% composed of alpha helices. So some proteins have no alpha helices whatsoever, and some proteins are made entirely of alpha helices. So for example, this protein here, which is ferritin, which is an iron-binding protein, it's made up about 75% of alpha helices. 25% of soluble proteins are basically composed entirely of alpha helices that are connected by loops and turns. So we'll talk about loops and turns a bit more in a little bit later. But this ferritin would be an example of that. So it has all these alpha helices, and then it's got some loops and turns that connect these different alpha helices to each other in the full three-dimensional fold. And then alpha helices are also commonly found in membrane-spanning proteins. So this is a receptor called a 7-transmembrane domain receptor or a G-protein coupled receptor. And you can see from the N-terminus to the C-terminus, it basically has a loop, alpha helix, through that spans the membrane, loop, alpha helix, loop, alpha helix, and it does that 7 times. So these alpha helices will span the entire membrane and be composed of hydrophobic amino acids on the side chain. So just a few more points about alpha helices. All right, so now that we've gone through alpha helices, we could talk about the other important secondary structure that we'll encounter, which are beta sheets. And so beta sheets are also caused by hydrogen bond interactions, but they're between 2 or more what's called beta strands. So this image here is of a beta strand, and it has this type of conformation where you have the amino group pointing over here. You have the alpha carbon with the residue pointed downwards, as you can see here. Then you have the carboxyl group, amino group pointed in opposite directions. Then you have this alpha carbon with the side chain pointing up, and that basically continues from there on out. And it has a very specific sort of measurements there. So between 1, 2, 3 different side chains will be a regular distance of about 7 angstroms. And importantly, these side chains, as I sort of already pointed out, are pointed in opposite directions from each other, as you can see here. So this would be a single beta strand. So beta sheets then are going to be caused by hydrogen bonding interactions between the backbones of adjacent beta strands. So this is different from the alpha helices, where it's basically the same alpha helix and then hydrogen bonding interactions within that alpha helix. So for beta sheets, it's this hydrogen bonding between the backbones of adjacent beta strands. So you can see here there is hydrogen bonding between this amino hydrogen and this carboxyl oxygen right here, and that continues throughout the beta strand. And so beta sheets can be formed by beta strands that are either going in the same direction or the opposite direction from that amino to carboxy terminus. So this here on the top would be an anti-parallel beta strand because they're going in opposite directions. And then down here would be a parallel beta sheet because they're going in the beta strands are going in the same direction. So again, sort of similar with alpha helices, the anti-parallel beta sheets are a bit more common in proteins than the parallel. And you can see because over the same difference, there's going to be a lot more hydrogen bonds at a closer distance than what you see for the anti-parallel beta sheet. And so you will find both anti-parallel and parallel in proteins, but anti-parallel are a bit more common. And you can also find mixed beta sheets that are caused by a combination of parallel and anti-parallel hydrogen bonding between those different beta strands, as you can see here. And as I said before, so multiple beta strands will combine to form beta sheets, but it's usually not just one beta strand, or I'm sorry, 2 beta strands or 3 beta strands. Usually it's like 3 to 7 beta strands will combine to form a bigger beta sheet. And these are depicted instead of as ribbons or cylinders, these are depicted as sort of flat arrows from the amino terminus to the carboxy terminus. So that's the direction in which they're pointing. And so you'll have these beta sheets here, and then the R groups, the side chains, will be sticking off on either side of this beta sheet, which is why it's so common, because it's so conformationally stable thermodynamically and chemically. And you can actually have proteins, it's very common actually to have proteins that are made up mostly of beta strands that'll form this, what's called this beta barrel region right here. So you can see this protein has beta strands, is basically made up of a beta sheet that forms this cylinder as depicted here and connected by those loops and turns, as just like we saw with the alpha helices. And so now that we've discussed alpha helices and beta sheets, we can talk about the third kind of secondary structure, which are loops and turns. And so if a protein is going to fold into a globular structure, it has to be able to reverse directions in the protein, and that is provided by these loops and turns. So they're located on the outside of the protein and are often involved with interactions with other proteins and the environment. And you can see here, these are also considered secondary structure, and they're also formed by hydrogen bonding interactions between adjacent backbone atoms. So you can see here, this protein is forming a turn where it reverses direction, and that's stabilized by this hydrogen bonding interaction right here. And here's a depiction of a beta sheet containing protein. And you can see this one beta strand is pointed this direction, and then there's a loop turn, and then it starts a beta strand in another direction to form this beta sheet. And these red regions, I believe here in this particular protein, are involved in binding of different molecules to that protein. All right, and so now that we've gone over alpha helices, beta sheets, and loops and turns, we can think about that Ramachandran plot, the plot that we talked about when we were talking about primary structure. So these beta sheets, alpha helices, and loops and turns are going to be formed by the local folding of those rotations about those phi and psi bonds, which are discussed in this Ramachandran plot. And then, of course, which is not really a big surprise, you can see that this region right here, which is very conformationally acceptable within the Ramachandran plot, this region right here that a lot of different of these angles are found in are what forms beta sheets. And then this region right here, which is also very common, these are the right-handed alpha helices. And then this little tiny region right here, which is less common, but still pretty conformationally stable in some instances, would be left-handed alpha helices. So this shows you how the primary structure and the secondary structure are linked. So the rotation about those phi and psi bonds at the alpha carbon, which is part of the primary structure of the protein, fold into these regular secondary structures, beta sheets and alpha helices, which are all accommodated for in the Ramachandran plot and are thus thermodynamically very stable. And that's why we find them so often when we're looking at protein structures. That's basically why they make up pretty much all of protein structures. All right, so that concludes lecture number 4, Protein Structure Part 1. And there's a few take-home points that I really want to emphasize. One is that you definitely need to know the difference between primary protein structure and secondary protein structure. So remember that primary protein structure describes the sequence of amino acids that are linked by peptide bonds into a polypeptide chain. You need to know that the peptide bond has some planar characteristics, whereas the other bonds in the polypeptide chain can rotate but have are limited by the torsion and the angles that they're able to make. And you need to understand that secondary structure describes the local folding of polypeptides into these regular structures. So you need to be able to recognize alpha helices, beta sheets, as well as loops and turns, and understand that these are formed by hydrogen bonding interactions on the polypeptide backbone. So I hope you guys all enjoyed the lecture, and please, as always, let me know if you have any questions.